Hello everyone, welcome to Evolution Part 2. In this video, we're first going to address how is evolution defined in genetic terms. We can say that evolution is change over time. It's species coming up and going extinct, but we can also define it in genetic terms. So the definition of evolution is change in the frequency of alleles in a population over time. So now you have to understand, what does allele mean? Allele means it's a variation of a certain gene which codes for a certain trait. So it's always a gene that codes for a trait, but there are different variations that allow us, to be different from each other. An allele could either be a dominant allele or a recessive allele for that trait. So for example, if you have the trait eye color, you can have the dominant allele that codes for brown eye, and you can have the recessive allele that codes for blue eye. The alleles are variations of the gene, and the gene codes for a trait. For every single one of us, as human beings, we carry 2 alleles of each gene within our somatic cells. So that means we can either carry 2 dominant alleles, we can carry 2 recessive alleles, or we can carry 1 dominant and 1 recessive. That will give us big A genotype, big A little a genotype, or little a genotype, as you can see on the bottom. So this distribution, as we remember in class, we wrote big A, big B, big B, little b, whatever. And we wrote out the genotypes of 10 different people. And then you can calculate the allele frequency, which we'll do in a second. But here we also have something called a gene pool. If we're considering all the genes, that means your hair color gene, your eye color gene, your height genes, your everything genes, we're considering all of those genes and all of the alleles of the genes of all the individuals within the population. That will be called a gene pool. For example, this right here will be called a gene pool. Now, allele frequency is the number of times an allele occurs in a gene pool. So for example, if we count all the little b's right here, 1, 2, 3, 4, 5, 6, 7. So we got 7 little b's in total. We use that divided by the total number of b's. 1, 2, 3, 4, 5, 6, 7. We have 7 pigs. That means there are 14 letter B's in total. So there are 14 alleles in total, but there are 7 recessive alleles. We do 7 divided by 14, which is 1/2 or 0.5. That will be the allele frequency of little b. So allele frequency is calculated as the number of alleles the question is asking for. It's either the dominant or the recessive divided by the total number of alleles. Make sure that you know within a population, if we have 10 individuals, that means there are 20 different alleles in total. So you will divide the whatever number that you count divided by 20. So now, this right here, you can see a change in allele frequency of a population over time. So at our original population has big A allele is 0.5, little a allele frequency is also 0.5. Because if you were to count little a, 1, 2, 3, we count in the letter little a, okay, not the individual. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. We do 10 divided by, so that 10 is the number of little a's, divided by 20, which is the number of a's in total, because there are 10 individuals, and each individual carry 2 alleles. That means there are 20 alleles in total. So that gives us little a equals to 0.5 and big A equals to 0.5. However, if the environment is only allowing these 5 rabbits to reproduce and all the other rabbits do not reproduce and survive, then our second generation of rabbits is going to look something like this. And you can see that there's a change in allele frequency. Big A allele frequency is now 0.7, little a is 0.3. In our third generation, because only those 2 rabbits reproduce, we have 1 and 0. So you see there's a change in allele frequency over time. This is called evolution. No, we're not. Okay. Now, the second one is 3 sources of genetic variation, right? We said mutation gives us genetic variations in the first place, but as organisms that can reproduce sexually, or even for asexual reproduction, there are ways that those variations in the genes can be shuffled around to allow individuals to have the same trait. So for example, if there's no sexual reproduction, when you were parents, when two parents have, let's say, 5 babies, these 5 babies should all have the same exact genes because they came from the parents, right? No, because there are other factors that goes into it other than just the mutation itself. So the 3 sources is mutations, genetic recombination, and sexual reproduction, and lateral gene transfer. Lateral gene transfer is also called horizontal gene transfer. So first off, mutations can be beneficial, harmful, or have no effect. Very few mutations can increase the individual's fitness, right? Because it's all by chance. Some of the mutations might be bad. Some of the mutations, actually a lot of mutations will have no effect, but occasionally those mutations can increase an individual's fitness, the ability to survive and reproduce. And then those individuals can pass on their genes and allow the allele frequencies of the population to change. Now, only mutations in germline cells can be passed on to the offsprings. So now, there's also genetic recombination in sexual reproduction. What allows you and your brother to look different from each other is because of sexual reproduction. So first off, during meiosis, there's a split of the homologous chromosomes and the sister chromatids, right? So that allows each gamete to be different from each other. And then fertilization combines 2 gametes together, one from mother, one from dad. And this allows you to have traits from both mother and father. There's crossing over during the process of meiosis as well. So we'll look at a picture of that. So that also increases the level of genetic variations. The last one is called horizontal gene transfer. Oh wait, lateral gene transfer is not actually during sexual reproduction. This is for asexual reproduction. Let's first look at meiosis. Fertilization is when you take 2 gametes and we form a zygote. So you should know a zygote is a result of fertilization. And the formation of a zygote through sexual reproduction allows individuals to have traits from both parents. However, the process of meiosis also creates a lot of variations. So for example, the part of meiosis called independent assortment allows homologous chromosomes to line up at the metaphase plate in different ways. For example, right here we only have 2 pairs of homologous chromosomes, and they all can already line up in 2 different ways. And if we have 23 pairs of chromosomes, homologous chromosomes, then there are 2 to the 23rd different types of lineups, which will give us 2 to the 23rd different types of gametes just from independent assortment itself. And then we have also crossing over. Crossing over allows homologous chromosomes, beyond the lining up, there is also crossing over happening to allow homologous chromosomes to exchange genetic material. So then the result is every single gamete is different from each other, which is why every single individual resulted in sexual reproduction, unless there's the possibility of twins. Everybody should be different from each other. So that's crossing over and sexual reproduction and independent assortment. Now the last one is called lateral gene transfer. This is how asexual reproduction can allow genetic variation. So asexual reproduction itself does not give rise to genetic variations because asexual reproduction means the offspring have the exact same genes as the parents, because they're asexual reproduction is that's how it works, right? The parents kind of make a copy of itself to have an offspring. However, organisms that go through asexual reproduction, such as bacteria, can do something called lateral gene transfer that allows genetic variations to be spread around. So how it works is that, let's say a bacteria right here, through mutation, can develop a plasmid that gives extra genes to the bacteria. So the bacteria has the chromosome, that's the genes that it originally carries, but then it can also have an extra piece called a plasmid. And this plasmid can be transferred from one bacteria to another. By 2 bacteria, when they're close together, they can potentially form something called a pilus, and this plasmid, this added piece of variation, can make a copy of itself and be sent from one bacteria to the other, or to another. So the result is both bacteria now has this variation. So why does this actually matter? So you know that bacteria can be resistant to antibiotics. Antibiotics are used to kill bacteria, right? But some bacteria through mutation, just random mutation, they happen to be resistant to antibiotics. They won't die when you take antibiotics. However, this is a real big problem because bacteria can actually spread this antibiotic resistant gene, which is anaplasmid, to other bacteria in the population to allow more and more bacteria to have this new variation, this ability to be antibiotic resistance. And this process is called lateral gene transfer or horizontal gene transfer. You can also call this bacterial conjugation. So lateral gene transfer has other methods, but this is only one of them. This is one's called bacterial conjugation. But either way, they increase genetic diversity for organisms that do asexual reproduction. Now let's talk about single gene trait versus polygenic trait. A single gene trait is a trait that's affected by only one gene and one gene only. Usually these traits, you can identify these traits when it's an either or case. You have very distinct traits. You can count how many different types of traits there are. And in this case, every single one that's listed on here is a single gene trait. So if you look at the definition of trait controlled by only one gene, the result is you have two distinct phenotypes, or a few more, but they're distinct phenotypes. You don't have anything in between. You have this, well, it's like A or B or C. There's nothing in between A or B. That's a distinct phenotype. And then, for example, color blindness will be a single gene trait. A polygenic trait is a trait that's affected by multiple genes, and the result on the phenotype of this is that you will have a whole range of traits. So instead of just having A, B, or C, you have A and B and everything in between, like A.1 and A.2 and everything, and all the way to B. I don't know if this makes sense to you, but anyway, for example, for height, you can have any kind of height, right? You can have any number. For example, in this graph, on the x-axis, you have 4 feet all the way to 7 feet, and every single number in between, 4.51, 5.3, 2, anyway. All of those are possible, and you're only able to have all these variations of traits, this countless numbers of variations of traits, when this trait is determined by multiple genes. This kind of trait is called a polygenic trait. So for example, skin color and height, those are typical examples of polygenic trait. Those are controlled by 2 or more, this right here should say genes, and then the result is you have a whole range of trait that's not an either or case, such as height of a human. So for example, in this case, if there is only white guinea pigs and black guinea pigs only, that will tell you that this is a single gene trait because those are distinct traits. You don't have, guinea pigs that are slightly gray, slightly even grayer, mostly white, those don't exist. That means this is a single gene trait. Now, the reason why we're talking about this is because natural selection affects single gene trait and polygenic trait in different ways. So first off, this right here shows you a single gene trait where you have 3 very distinct phenotypes and you don't have, maroon colored lizards. And because you have these very distinct phenotypes, over time, the distribution and the frequency of each phenotype can change. Such as this, from the initial population all the way to generation 30, you can see that now you have 40% to 60% rather than 80, 10, and 10%. So single gene trait, for a single gene trait, because it's only one gene, meaning you only have 2 alleles, the allele frequencies will change, and the result is the phenotypic frequency also change. However, for polygenic traits, because you start with a curve like this, where the x-axis will be the trait itself, the whole range of traits. For example, in this case, we have finches and the depth of the finch, how wide it is, all the way from 6 millimeters to 14 millimeters. And then on the y-axis is basically how prevalent this certain trait is. So, for example, if we were to look at finches with 9-inch beaks, we have about 50 individuals of finches that has this particular beak depth. Another example could be, let's see, 12 for 12-inch beaks, 12 not inch, 12 millimeter beaks, we maybe only have 3 individuals that have it. So the x-axis is the actual trait, the y-axis show you how, like, the taller the bar is, that tells you more individuals would have that particular trait. So natural selection on this polygenic trait, because there's so many different types of that one trait, you can't just have allele frequency change of one gene, right? There are many genes that's affecting this trait. So, so how it works is that you will look at the environment and see, out of this entire range, 6 to 14, this entire range, what type of trait is more fit. Okay, so maybe it's smaller beaks that's more fit in this environment, the medium or the large, but it all depends on the environment. So here are 3 possible outcomes. The first one is called directional selection. This is when, so we have our original curve right here. Directional selection happens when only the either extremes, so either this extreme, the end of the curve, or this extreme is the most fit within the population. Okay, so for example, we have these lizards, and having long tails for these lizards is good for its survival because it scares away the predators. So then the result is, over time, a lot of the really short tail lizards would die, and the longer tail lizards would be better at surviving and reproducing. So the result is this entire curve of distribution will move this way, where originally you have mostly the most of the population would have medium-sized tails. However, over time, the medium-long tails will be the most prevalent within the population. The frequency of the medium-long is the highest. So this is called directional selection because, you know, within our whole range, right? Let's say 4 feet to 6 feet, whatever. Either end is better for survival and reproduction, and the curve is going to shift toward whichever way that allows the individuals to be more fit. So in this case, having longer tails is more fit, so the entire curve is going to shift this way. Now the second one is called stabilizing selection because it's stabilizing the curve. We start with a curve that's you know, pretty, we have a pretty big range, and we have the medium size, whatever is the most fit, so you have the most amount of it. But if the environment allows the medium size to be the most fit, then your curve is going to narrow because being on the 2 ends is not very fit, but being in the middle is more fit. So then the middle guys are going to survive and reproduce more. So then your curve is going to turn into something like this. So the curve is going to narrow, and the curve is going to go higher. This is called stabilizing selection. For example, having short tail or really long tail is not good for these cats. So having medium tail is the most fit, and the result is the curve is going to go up. This is called stabilizing selection. It says selection against both extremes. So when we say select against, that means those traits are not selective for. They're not good for the survival and reproduction of the individuals. The last one is called disruptive selection because it disrupts in the middle. Okay, so we start with a curve like this. However, this end and this end allows the individuals to be really fit, and in the middle makes them less fit. So over time, you would have more and more of the 2 extremes and less of the medium. So it says selection against the mean, right? So the medium is the mean, or the, you know, the average. Now we can quickly click through the notes. Directional selection, cause, the effect, make sure you read through this, okay? We have individuals at one end of the curve has higher fitness, so this curve is going to shift. For example, average beak size. Well, the result is the average of the overall population is going to, you know, shift to that end as well, either getting bigger or smaller. Here's directional selection again. Stabilizing selection. The effect is more of the population will have the close to average, so then you will have more and more of the average, right? The longer or the higher that part of the curve is, the more individuals, or the more percent of the individuals will have that particular trait. The last one is our disruptive. The result is you would have single curves split into 2 curves in the middle. All right. Now, the last part is, second to last part is genetic drift. Genetic drift is a random event that causes a change in allele frequencies. So natural selection is when the environment is determining certain individuals within the population is more fit than the other ones. However, genetic drift is all random. Okay, it's not dependent on the environment. There's nothing about you that makes you more fit. It just happened. You're the lucky guy. Okay, so how would this work? Genetic drift, first off, is a chance event that causes change in allele frequencies within a population. Chance event means it's random. Natural selection is not random because the environment does determine certain individuals to be more fit and to be able to, again, survive and reproduce and pass on their genes. So the first type of genetic drift is called bottleneck effect. Bottleneck effect happens to a population where you have an original population, but then unfortunately there's some sort of natural disaster that kills most of the population. And the few individuals that are left over are going to have a different frequency of alleles compared to the original population, right? So if you just look at this bottle right here, if you pour a few of the marbles out, the frequency of the marbles is going to be different later compared to the original. So a dramatic reduction in the size of the population caused by a chance event is called genetic bottleneck. So for example, if you have an island, you have these beetles. Why are beetles so sad? Let's say you have on the island you have turtles, and these turtles. How about mushrooms? They're these mushrooms. Mushrooms don't move, but they also have variations, right? And this volcanic eruption killed a bunch of mushrooms because that's what volcanic eruptions do. And the leftover mushrooms are going to have different allele frequencies compared to the original mushroom population before the volcanic eruption because a lot of them died, and the leftover ones are not, did not survive because they're better at surviving. They survived because they were lucky. And in this case, they didn't get run over by the volcanic eruption. So this is called bottleneck effect. The founder effect, so bottleneck effect is one population, right? And it's reducing by a lot. The founder effect is you have one population that's original, and then you have a new area that's not founded by this original population yet. So we have this original population. Some of the individuals, very few individuals, wandered over to another place and kind of started their own new population. So the result is this new population founded by a few individuals from the original population, they will have a different allele frequency compared to the original population. So that is called the founder effect. Okay, so for example, right here we have our ancestral population of butterflies with different variations. It just turned out that the maroon butterfly, well, two of those, and one orange butterfly came over to the new area and they started their own new population. Then the allele frequencies of this new population will be different from the original population. This is called the founder effect. This is also not done by natural selection. These few individuals not necessarily can survive and reproduce better than the original ones. They might, but they also might not. It's all random. However, there's still a change in allele frequency over time, which is called evolution. So not only natural selection can result in evolution, genetic drift in two different ways, both bottleneck effect and founder effect, can result in evolution. So the last one, yay, finally, we have Hardy-Weinberg equilibrium. What is Hardy-Weinberg equilibrium? It is when the population does not change its allele frequency. And we want to think about what are factors that can or cannot change allele frequencies. And if it doesn't change the allele frequencies, we call it Hardy-Weinberg equilibrium, okay? So the Hardy-Weinberg principle states that allele frequencies in a population should remain constant unless one or more of the factors cause those frequencies to change. And then we're going to look at the factors. So if your first generation looks exactly like the second generation and the allele frequencies are exactly the same, we say this population is staying in Hardy-Weinberg equilibrium. Now, here are factors that can disrupt equilibrium, meaning that if you have the opposite of the factor, that means your population will stay in Hardy-Weinberg equilibrium. You will not change. So here's the first one. Okay. Non-random mating means not every individual is mating with, well, has the ability to mate with individuals, right? Or this can also be called sexual selection. So during sexual selection, let's say birds, for example, we have birds that are really boring looking, usually the female ones, and birds that are really extravagant, such as the male ones. And the reason why male birds are often prettier than the female birds is because females are, in bird populations a lot of the times, are doing the sexual selection process where they would only select individual males with certain traits, usually the prettiest, the most colorful male birds. The female birds will select those for reproduction. So this is called sexual selection, right? So if sexual selection is happening, that means not every male bird is going to survive and reproduce because they're, some are selected and some are not. This will disrupt equilibrium because those that did not get selected to reproduce will not pass on their genes. The second one is called small population size. Whenever you have a small population, genetic drift in the form of either bottleneck effect or founder effect are more likely to happen, or it's going to have a larger effect, right? If you think, let's say we only have 10 beetles, we're just going to keep on using beetles. If we have 10 beetles and some dramatic event kills 5 of those, for example, the allele frequency is going to be quite different after the genetic drift event. However, if you were to have 1,000 beetles, killing 5 of them is not going to be as big of a problem. So a small population results in an easier change in allele frequencies. Now, the third one is immigration or emigration. You have a population, some comes in, some individuals come in, some individuals come out. This process is also called gene flow. This will definitely disrupt equilibrium because those that come in will bring new genes and new alleles to the population. Those that go out will take the genes and alleles with them, which is going to change the equilibrium. Next one is mutations. Obviously, mutations can introduce new alleles. That's going to change the allele frequency, which disrupts equilibrium. Last one, natural selection, that we spend a lot of time talking about. Natural selection allows certain individuals to be more fit than the other ones, which means those individuals can survive and reproduce and pass on their genes more often than the other ones. So the result is the allele frequencies over time is going to change. That's the end of the video. And here's Marshy, hoping you'll do really, really well on your test. Yay!